So section 15.7, specific immunity and vaccination. The learning outcomes we have for this are to list the 4 categories of acquired immunity and provide examples of each, and then to discuss the qualities of an effective vaccine and list several types of vaccines and how they are utilized today, and then explain the principle of herd immunity and the risks that unfold when not maintained. Okay, so 4 types of acquired immunity. These are based on what you receive, so like and how it is given to you, how you receive it and what you receive. And so first of all, if it is natural, it's going to be acquired through normal life processes, like biological processes normally happening without any medical intervention. So normal life experiences not induced through medical means. That would be natural. And if it's artificial, that is going to be purposefully given to you, and it is through medical procedures. And if it is active, you're receiving the antigen, and that causes you to create memory of the event. You get an immune response and you get memory. So it's going to be lasting. Where if it is passive, then you get the antigen, or sorry, you get the antibody. You get the antibody and you have no memory. So there's no response. You're just given the antibodies. They were produced outside of your own body. And so those are the 2 things. So if it's active, you are given an antigen. If it is passive, you're given an antibody, no memory, and this is going to be short term. So sometimes that's necessary because you need to get it. You don't have time to wait for it to produce the antibodies yourself. So you need it right away. So some examples, like this person here is obviously ill, and so illness is natural, and you come into contact with this, you produce an immune response, so it's active. So natural active would be illness. Natural passive would be from mother to child, and that can be through the placenta or breast milk. So artificial active immunity then would be vaccination, that we kind of typically think of in a vaccination, and that can either be through a shot like this, or it could be something like an oral. So there was the oral polio vaccine that was given through the mouth, and then a lot of times it's through mucous membrane, so it's like through the nose. Passive immunity, this is usually by IV, and this is just antibodies given to the person, and this would be like immune serum globulin or specific immune globulin, and we're going to talk about these. So this is like immune therapy. Immune therapy is the term that we would use for that. So vaccination is the active, and immune therapy through some sort of IV usually. It's delivered through an IV of some sort. So let's talk about these 2 in particular. I think it's pretty obvious, like what is illness? And passive immunity from mother to child, those are fairly self-explanatory. So let's talk about the passive, artificial passive, and artificial active. So artificial passive immunization is an intravenous immune globulin. So gamma globulin is also called immune serum globulin. Immune serum globulin. So you're getting IgG antibodies, and that's why it's also called gamma globulin. But it's extracted from a pool of human donors that are from a particular area, and they will give this to military personnel that are being deployed, because they may not have been exposed to the pathogens, the potential pathogens that they're going to be exposed to. They've never been exposed to them, so they don't have any immunity, and that can be a mission aborting kind of thing if you get all these people sick. So they give them a pool of donors, a pool of blood from human donors in that area. And so most of the forms are injected intramuscularly, and it gives you about 2 to 3 months protection. So this is pretty short-lived. So it's less than a year. It's just as long as the IgG antibodies will last. So about 45 days would be an IgG, and then 2 to 3 months, that might be about all you're going to get. So with a specific immune globulin, so this is the SIG. This is from a more defined group of donors who are convalescing, and they're in a hyperimmune state. That means people who have recently had COVID, they were giving their blood, and the serum was being, they were taking the serum off of that and giving people that to treat COVID. So before COVID, there was Ebola, and there was a doctor and a nurse who survived Ebola because they were given this specific immune serum, specific immune globulin. And so it has a high titer specific antibodies that are for the particular disease that they were exposed to. So it's given in prophylaxis in patients who have been exposed or may be exposed. And it's just really limited. It's limited by, how many surviving patients do you have? If everybody is killed off, you have nobody to get this immune serum from. But, if you have 65% mortality, that means there's still 35% of the people that survive. They have antibodies to this, and you can give people that specific immune globulin if they have been infected. So it's very useful in some cases. So the artificial active immunity is vaccination. So the last two things we were talking about, those were passive. You're just being given antibodies. In this, it's artificial active. So you're being given antigenic material that is antigenic. So you're given antigens, and they are antigenic. They can create an immune response, but they're not pathogenic. So again, they're antigenic, but not pathogenic. And so the discovery of vaccination was the farthest reaching and most important developments probably in medical science. The very first person to use this was Edward Jenner, and he gave it to a young man whose name was Joseph Phillips, or Phipps. Anyway, that was back in the 1700s. So the very first vaccination was with smallpox in the 1700s, and people thought they were going to have cows growing out of their arms because of that. So Edward Jenner was the one who made this against smallpox. So why do we call it vaccination? Well, because smallpox is variola, and before, that is the name of the virus, and before this procedure was developed, they would do a thing called variolation, which was making a little cut in your arm and then just slathering it with the pus from the smallpox. But then they found out that cowpox would give you immunity without making you sick. So this variolation was very damaging, and you actually got the illness, and just a mild case of it because of where it was delivered. So it had a different portal of entry. We'll talk about that in chapter 13. But because it was in the arm and not being breathed in, which is the normal portal of entry for smallpox, it did not make them as sick. but they still got sick. They still produced the little poxes. So cowpox is the vaccinia virus. And that is where we get the name for vaccination, because you're being given cowpox. And then that doesn't give you the pathogenicity, but it does have the antigenicity of the smallpox virus. And so it stimulates a primary immune response. You're basically priming the immune system for a future exposure to the pathogen. And it gives you, when the pathogen later enters the body, you get a powerful secondary response that is immediate and robust. It's powerful and it's sustained. And so that is the point and purpose of vaccination. So the principles of vaccine preparation. How do we make the vaccine, and what do we look at when we're trying to consider the production of a vaccine? So especially when they were looking at COVID, they had the code for the virus early on, within a month after the first few cases had been identified. And so they identified the antigen, which was a spike protein in the surface of the virus. And that is a lot of times what we make the vaccines against, because it's what our immune system detects. It's the thing that's sticking out and that it's going to be used to get into your cells. And so we want to stop it from being able to do that. So we want to know that it's effective. And that was one of the amazing things about the COVID vaccines. They were very effective, like 95% effective. We want them to be easy to administer. We want it, if it could be administered by just eating something or drinking something, that would be great. But a lot of times it has to be in a shot of some sort. Safety, we want to make sure this is safe, but it's not going to harm the patients who are getting the vaccine. And we need it to be cost effective. And so all of these things were kind of taken into consideration when they developed the vaccines. So the requirements, we want it to have a low level of adverse effects. We don't want it to cause serious harm. It should protect against the natural wild forms of the pathogen, and it should stimulate the antibody and the cell-mediated or T-cell response. It should have lasting long-term effects, produce memory. It should not require numerous doses or boosters. And it should be inexpensive, have a relatively long shelf life, and easy to administer. So not all of these. There's no perfect vaccine. And you can probably point out some things that did not happen with the COVID vaccine, like it wasn't as easy to administer. It did not have a very long shelf life. I don't know how expensive it was to make it, but these are all these things. This is what we ideally would like to see in a vaccine. So the whole cell vaccines, they're what I call the first generation vaccines. These were some of the first vaccines that we were able to develop, and they're basically two types. So the first type is the killed or inactivated, and then the second type is the live attenuated cells or viruses. So they've been weakened. And because they are weakened, they are still able to give us lasting protection, but they require fewer doses and boosters. And so they're very effective against at inducing cell-mediated immunity. So those are some good things. The benefits of having a killed vaccine is there is no way to get the disease. And so that is a positive kind of attribute of the killed inactivated vaccines. The weakened, there's a possibility of getting the disease. And so that is obviously a negative aspect of it. So the possibility of getting the disease, if you are immunocompromised and this is a weakened, but it could still cause disease in you, then that's not good. But again, the benefits are that they require fewer doses, boosters. You don't have to have a booster. And but both of these do give you long-lasting protection. So the bad thing, well, not bad thing so much, but a negative for this would be that you do require boosters for this killed vaccine. Because they're not able to replicate inside the host. So these replicate inside the host, and so they don't need to have a booster because they continue to produce antigenicity, and they continue to produce a good immune response. So there's one positive and negative for each of these, at least one positive and negative. But I would just say that both of these give us long-lasting immunity. And long-lasting because it is more than a year, at least a year. And we see this with the killed and with the attenuated influenza vaccines. So it's not a short-term fix. You know, the short-term fixes be the immune serum globulin, where you're just giving antibodies, or you know, breast milk that just gives antibodies. So you're actually developing memory cells for this, even though you're not getting the disease. And so those are the two types of first generation or whole cell vaccines. So the subunit or second generation vaccines, you are getting a piece of the bacteria or the virus. And so there is, the benefit of this is that there is no chance of getting the disease. No chance whatsoever of getting the disease. Because you're not even given the whole organism. You're just being given a part of the organism. So that is definitely a plus. And they're not terribly difficult to necessarily prepare, and you don't necessarily have to know how they're prepared. But the just part or subunit, that's the second generation. And then genetically engineered, that would be a third generation in my estimation. So toxoid preparations would still be kind of the second generation, but genetically engineered, that would be a third generation. And so how we make those is how we make these, that's going to make the difference between a second and third generation. But genetically engineered, we're going to call that third generation. So new vaccines, they're trying to make new vaccines for all these different viral diseases especially, but some other diseases that are affecting people in third world countries, especially diarrheal diseases and worm infections, malaria, HIV, AIDS. So all of these would help the people from getting these diseases in countries where they don't have a really good healthcare system. So that's going to be really important. And then some various respiratory diseases. But most existing vaccines are out of reach for much of the world's population. So it's really hard to get them and deliver them to them. So we've still got a lot of challenges ahead of us. So the third generation DNA vaccines, you have a microbial DNA is inserted into a plasmid, and it serves as a vector. That means that's the method of getting it into the cell, and that's inoculated into the recipient. So we put it into a bacteria cell, or we just give them the plasmid all together, and then the plasmid is taken up by the cells, and then they produce the antigenicity, the subunit, the piece of the virus or the bacteria cell, and sometimes it's presented on the surface like this. So humans take up the plasmid somehow and express the microbial DNA. So it's using the protein synthesis machinery of our cells to make those subunits. That's how we're making the pieces of the virus or the pieces of the bacteria cell. So the foreign proteins will be recognized by the immune system, and it sensitizes the immune system to make the B cells and T cells be primed for this particular antigen when you come into contact with it, if you come into contact with it. So I would pose that there's a 4th generation DNA vaccine. Well, it's actually not a DNA vaccine. That would be the mRNA vaccines that we developed for COVID. And so that will be in another video. But I believe that this is going to be a big part of a new edition of the textbook, is all these different vaccines that they've made for COVID, which is an mRNA vaccine. So it's made really very differently. You don't have to put the DNA into a vector, but they do have little fat lipid nanoparticles. So balls of fat. They put the mRNA in balls of fat, and then the balls of fat are injected into you, and the fat helps to transfer, get it across the membrane into your cells, and then your cells make the antibody, or the proteins, like the spike proteins, and then secrete those, or yeah, they're made, and then that is going to induce the immune response in the cells. And it's very effective. I mean, the 2 major mRNA vaccines were 95% effective, 90 to 95% effective, depending on, how long it was after the vaccine was given. So how do we administer them? Subcutaneously, intramuscularly, intradermal. There's also oral or nasal vaccines that are available. So these are the traditional ways, and probably the most common is the intramuscular, intradermal. So they a shot in the arm. And sometimes they'll add an adjuvant. So you may have heard this term before, but may not have understood what it means. And so basically it's a special binding substance that enhances the immunogenicity. Like aluminum seems to be an adjuvant that's very effective. And it helps to participate. It's like that carrier protein for the hapten. It makes it more antigenic, makes it recognizable by the immune system, and it precipitates the antigen and holds it into the tissue so that it can be released gradually. So there's a lot of different vaccines that are now being given an adjuvant to help give it a better efficacy. So side effects we see are localized reactions at the injection site. That's pretty common. Might have a little redness or maybe a little bit of achiness right there at the site of the injection. Fever and allergies. Some people are allergic to parts that are used to make the preparation, the vaccine. And so some of the common side effects are just a local reaction at the site of the infection. So some of the common side effects are a local reaction at the injection site, fever, allergies, and rarely you'll get these other side effects like panencephalitis, which is an infection that happens with measles vaccines, and you get about 1 in 220,000 of the vaccines will result in this swelling in the brain. Back mutations can occur. This happened with the polio vaccine, and a few people would get this every year. So it would back mutate to the original virulent strain, and it would give them polio. Neurological effects of the unknown cause, and so pertussis and the swine flu vaccine, they kind of took off the shelf because they were causing some unknown neurological effects. And allergies like to eggs or tissue culture ingredients are something that also happens. So I'm not saying that vaccines are perfectly safe and 100% nobody is going to get these side effects. It does happen, and that's why they give you the list of all the side effects, and you need to know whether that's something that you might have a problem with, especially like if you're allergic to eggs. So some people have attempted to link childhood vaccinations to the development of diabetes and autism and asthma. And there's been a very public debate about this over the last 20 years. And it's very difficult to discriminate, like what's good information, what's bad information. And wow, do we know that after COVID? That is for sure an issue. especially with the advent of social media. Anybody can say anything and they don't know what the heck they're talking about. And so in 2006, they definitively came up with the, no, MMR vaccine does not cause the autism. And that was after reviewing many, many studies. So herd immunity is something that you need to understand as well. The reason that vaccines are so important is because if you have a herd immunity, then you break the chain of transmission in the community. So if you have a sufficient number of immune individuals, then the microbe cannot spread to them and they can't spread it to other people. So the collective immunity through mass immunization confers indirect protection to non-immune members. And that is right there, that is kind of the definition of herd immunity. So collective immunity through mass immunization that confers indirect protection to non-immune members. In other words, everyone's vaccinated and therefore they can't give you the disease and they can't get it, so they can't give it to you. And so you have to maintain this certain level of herd immunity through immunization to prevent epidemics. So when they have had lower numbers that they have the herd immunity levels have gone below the threshold, then we see outbreaks. This happened in the Bronx with measles, mumps, rubella, especially with the measles. And so we've seen outbreaks over the last few years of this. We talk about herd immunity with COVID a lot, and where we need to have like 75%. That is not just a number that they picked out of the air. It's based on the R0, which is the reproductive rate of the vaccine. There's just science behind it, is my point. It's not just a random number. And to this date, we still have not reached herd immunity. There's about 69% vaccination as of today, which is July 19th, 2022, when I'm recording this. That is where we're at right now. And some places have less than others. This is for the whole United States. And that is just not enough to prevent the outbreaks. And so we're seeing another spike in cases here in mid-July of 2022. So will we ever have herd immunity with this? I don't know. It constantly is changing, and one of the things that you have to understand about COVID is that if we're going to prevent further variants from forming, you've got to stop the chain of transmission. And the longer that we go without having herd immunity, the more variants will come about, and even people who are protected now are getting it. So even people who have had immunity, they're going to need boosters. And so that's going to be a thing for a long time. We're going to have to probably have a flu and COVID vaccine to maintain the herd immunity, or to obtain some sort of herd immunity. So who is vaccinated and when? And so traditionally we vaccinate in childhood. Adults, though, will oftentimes need boosters, and they'll need some things for that are necessary for adults, infections such as pneumonia, shingles, things like that. So even if you've had chickenpox as a child, it might be good to have a shingles vaccine. And pneumonia is one of the #1 respiratory illnesses that kills people in the 55 and older category. So here are a couple of typical vaccines that are given, and just between the 1 month and well, even at birth to like 18 months, you have quite a few vaccines here. So hepatitis B, DTaP, HIV, PCV13, which is the pneumococcal vaccine, IPV inactive polio vaccine. The influenza needs to be done yearly, MMR, varicella, which is chickenpox, and hepatitis A. So they've made a lot of these in combinations, like there's a quad vaccine that they're giving that has several of these things. I think it's MMR and varicella or something like that. So these are some of the typical ones as of 2019. There's also a list, and it's in your book, of some of the vaccines that adults need to get, and adolescents. So you need to keep up with your vaccines and know what you are and are not vaccinated against. If you're going into healthcare field, there are going to be many vaccines that they are going to require, because you're going to be in contact with people, and it's not just for you, but also for the hospital community, so that you're not spreading it to people who are very immunocompromised when they're in the hospital. So concept check 15.7. Breastfeeding is an example of blank blank immunity. Vaccination is an example of blank blank immunity. Vaccines are either blank organism or part of organism preparations. And there are vaccines against all major infectious diseases. True or false? Blank blank, blank or blank vaccines stimulate IgA production on mucous membranes. And there is no link between MMR vaccine and autism. So let's answer these questions one at a time. So the breastfeeding is an example of natural and passive immunity. So you're given antibodies, and it only lasts as long as the antibodies last. So as long as you breastfeed for however long after, about 2 to 3 months after breastfeeding, that's about as long as you're going to get that colostrum, the benefit from the colostrum. Vaccination is an example of active and artificial immunity, because it's not a natural process and you're given antigens. So the antigens create an immune response, you have memory of it, and that's what gives this type of immunity. So vaccines are either blank organism, so whole organism, or part of organism preparations. And true or false, there are vaccines against all major infectious diseases. False. There are not, obviously. Otherwise, we would have defeated all disease. So blank or blank vaccines stimulate IgA production on mucous membranes. I would say those are probably the weakened or weakened, attenuated vaccines. Nasal, I'm not sure what blanks they're probably trying to put in here, so that I'm not sure about. And there is no link between MMR and autism. So they have looked at thousands and thousands of people through many, many different studies and cannot find a causal link between MMR vaccine and autism. And so unfortunately, they spent many, many years studying this, going down the wrong path, and now they still don't have like a cause for autism. So it is difficult, I understand, because you don't want to give your child something that's going to cause them to have autism. But I can just tell you the Medical Association, the Journal of the American Medical Association, published this in 2006. So it's been a while, and they have not found any link to MMR and autism. So that is all I know about vaccines. There's a lot more to discuss about the COVID vaccines, and for that I will have another video and audio video for that.